So we're ready to go here. We are still clearly working with some minor adjustments relative to attempting to get Ian McDonald and Miguel Seabra to zoom in live. That might happen. If it does, Dr. Viki Kalatzis has graciously agreed to work with us from a flexibility point of view. If they're not able to zoom in functionally, then Dr. Viki Kalatzis is all set to handle the entire presentation. So right now I'm going to just quickly introduce Beth Voss, and she's going to tell you a little bit about Miguel Seabra and Ian McDonald.
Oh, that's not necessary. Hello, everybody. I don't think these two gentlemen need much of an introduction, but we're going to go ahead and introduce them anyway for the folks that are new. We have two really pivotal foundational cornerstones of our organization, which would be Ian McDonald and Miguel Seabra. And due to the internet, as Neil said, we're trying to get them live to be able to come in. And it's just, internet. We know Zoom, right? Everybody's experienced that this year. So a little bit about Ian McDonald. Ian trained in genetics, first at McGill, then in Ottawa, where he met a family with choroideremia who asked him to undertake a research project on mapping the gene, and that was in 1983. He continues to work with patients and families with choroideremia and collaborators across North America and across the globe. He has been very thankful for the support he gets from the CRF in the USA and Canada for his lab. And the final comment, and just so that everyone knows, Corey McDonald is definitely the son he never had. That was written by, of course, him. Yeah, that wasn't written by Corey, no. Dr. Miguel Seabra, MD, PhD, is a leading researcher in biomedical sciences. He is the coordinator of a pioneering clinical trials of retinal gene therapy and a founder of Nightstar Therapeutics. With early support from the CRF, Dr. Seabra was responsible for creating the first mouse model for CHM in the early 2000s. So I think if we just give him a little bit of both of them, round of applause, that'd be awesome.
Thank you. All right, it's my pleasure to introduce Dr. Viki Kalatzis. She has taken the time and effort to come here from France. Obviously, travel is more difficult these days. So Dr. Viki Kalatzis, she works out of the Institute of Neurosciences in Montpellier, France. And she heads up the vision team where they work on a variety of inherited retinal diseases. And I will let her tell you more about the work she's doing. And please bear with her. She's working on a technical balancing beam here. So thank you, Viki, and we greatly appreciate your providing us information and updates.
Hi, I'm Viki. I work in France, as Neil said. I've been working on choroideremia. When I started working on retinal diseases, choroideremia is the first disease I started working on. So it's my favorite, if you like, because I've really been on it for the longest now. So just in case you're wondering, since I'm at it, I'm Australian. I'm from Greek origin. That's why I have a strange name, and I work in France. So I can't see what you have there. I hope you have a title slide. So today was supposed to be 3 people talking about what is choroideremia, where have we been, and where are we going. And so I'm going to try and do this by myself. I have always considered Ian as the grandfather of choroideremia and Miguel as the father, so I'm a poor substitute, but bear with me. So I will begin as an introduction, though, into the retina and the eye. And the retina is the tissue that lines the back of the eye, and it is composed of layers, of 3 layers of neuronal cells. And behind these neuronal cells is what's called the retinal pigment epithelium. And this is a tissue that really its role in life is to help the photoreceptors survive. And so when light enters our eye, it is projected over the whole retina, and it goes right through the retina to the back where the photoreceptors are, and these are our light sensing cells. And these cells will take the light signal, they will transform it into an electrophysiological signal that can then be sent by the other cells to the brain. And this way, the brain receives a signal that it can interpret. And we have two types of photoreceptors. There are what's called rods and cones, and these have different roles. So the rods are the cells, they're more in the periphery of a retina, and they are there to absorb light under low light conditions. So they contribute to our peripheral vision and our vision at night. Whereas the cones, they're in the center, and they're responsible for all our color vision, our fine vision, our day vision. And in the retina, we have a lot more rods than we have cones. Cones are a lot more sensitive to light. So inherited retinal diseases, they actually are a very big group, of which choroideremia is one disease. And these diseases are characterized by a loss of photoreceptors, a gradual loss, and also the supporting tissue. In fact, both can degenerate in the disease. And we divide these diseases into mainly two large groups, and they're based on which type of photoreceptor is lost first. So if we lose the rods first, we will lose our peripheral vision and have night vision. And if we lose our cones, we will lose first the central, the fine vision. And so there's a very big group of disorders. They're very different, all of them. They can progress at different speeds and different severities. So there is a very big clinical heterogeneity. And they are genetic disorders, and they can be due to mutations in genes of the photoreceptors or of the retinal pigment epithelium. But it doesn't really matter, because if one of these, they're really a couple and they rely on each other to live. So if one of them dies, the other one will die. And there's also a very big genetic heterogeneity, because there are over 270 genes that can go wrong and that can give us a retinal disease. And these, when we talk about genes and mutations, we can also have them go through the family in different ways. So for example, I think as most of you know, we always have 2 copies of each gene. And if we have mutations in both copies, these give what are called recessive disorders. If we have mutations in one copy on the X chromosome, these give X-linked disorders, and it's usually the males that are affected. And we can also have what's called dominant disorders, where we can have just one gene that's mutated, but it's going to affect the other gene and prevent it from working normally. So this is the basis of all retinal diseases. But it's choroideremia that interests us here today, and choroideremia is due to the CHM gene, which is on the X chromosome. So it's called an X-linked disorder. And choroideremia is characterized by the loss of the RPE, this supporting tissue, and the photoreceptors, the light-sensing tissue. But it's a little bit different from other retinal diseases in that there is a third tissue that is affected. And this is a vascular tissue that's underneath or behind the retinal pigment epithelium, and it's called the choroid. And the choroid actually brings all the nutrients towards the retina, and it takes away all the waste that we don't want. And so in choroideremia, it's these 3 tissues that are affected. And the gene is actually expressed in all of them. It's actually a gene that is expressed everywhere in the body, in every cell. But it is only the retina that is affected when there is a mutation. And so what happens? So as I said, is X-linked. So females are what we called carriers. And so if you look at the eye of a female, it generally looks, the back of the eye looks red, healthy, like a patient without, like a person without a mutation. We can just sometimes see small spots on the retina. However, as the disease progresses, and because the choroid is also degenerating, we actually have an unusual aspect of the cornea, of the back of the eye, and this is because the retinal pigment epithelium is pigmented, the choroid is pigmented. So when we lose all the pigment and we take a photo of the back of the eye, we don't have the same reflection of the light, so the back of the eye is no longer red, but it's yellow. And so this is something that it's very particular for choroideremia. I will also add that in addition to all the difficulties today, the slides on this are very small and I don't have my glasses on. So it's also another challenge. So bear with me. So to begin, I wanted to talk about the CHM gene, but just to explain in case there's anybody here that's not really familiar with genes. So a gene is just a piece of DNA, essentially, but it is made up of different subunits. It's made up of things that we call coding exons, and it's made up of sections that we call non-coding. So what does this mean? Essentially, for me, a gene is like a book. It is just sentences. And the cell has to be able to read this book. So she will start reading and everything will be fine as long as she's in a section that makes sense in the exons. But then she'll get to a section that no longer makes sense. The sentences don't make any sense. And the cell cannot read these. No, thank you. He just gave me glasses. The cell cannot read these. So what the cell has to do, the first thing before she can actually do something with a gene, is she has to rip out the pages that don't make sense. And that way she will have a page that makes sense, followed by a page that makes sense, and she can read a story. And ripping out these pages that do not make sense is called splicing, and it gives what we call an RNA. So after COVID, everybody knows now what an RNA is. And an RNA is very important because it is just an intermediate step to give a protein. And the protein is the story that the cell has made something with, because and that is what plays a role in the cell, and that is what is important. And so Rep1 is the protein coded by the CHM gene. And so when we have mutations, we I will use your glasses for a second. Thank you. When we have mutations, we actually have like a spelling mistake in one of the sentences, even in the region that normally makes sense. And that is what the cell has problems with. And generally what the cell will do will say, I can't read this, I can't make sense of it, I'm not producing it. And that is what is the case with most of the mutations due to CHM. So there's a lot of different mutations and they do different effects within the cell to the gene. But there are big classes, and the main classes in CHM are what we call loss of function mutations. So they're the ones that the cell really can't read and says, I'm not making any protein, and so there is no Rep1 protein made. There is a second class of mutations, which is the same. The result is the same. There is no protein made. But I just want to mention them because they're important for the talk later. They're what we call nonsense mutations. So what they mean is there is the change of one little letter, and it actually what it does is it introduces in the DNA like a stop sign. So it's called a premature stop because at the end of all genes, there is always a stop sign. It's like the end in the book. The cell can get to the end and know it's the end and know she can stop reading. And what these mutations do is it puts the stop sign earlier. So the cell will get halfway through the book and will think she's reached the end of the story and stop there. And these nonsense mutations are actually very, very common in choroideremia. They're about 30% of patients. And as I said, the end result is still no protein. And then another class is also interesting, and this is splice site mutations. And what these are, just checking I'm in the right category, yes. What these are is essentially, there's also just a little letter change. But what it will do is the cell, instead of ripping out the pages that just don't make sense, she'll take some pages that do make sense with her. And so she will remove exons. It's called skipping exons. And so in the end, it's the same result. She doesn't understand the story of the book and she doesn't want to do anything with it. So what does Rep1 do? And thanks to all the seminal work of Miguel Seabra, we have a very good grasp of what Rep1 does. And it's called Rep escort protein. So this protein really is just a chaperone in the cell. You can think of it as a nanny. And what it does, it goes around the cell and it takes children by the hand. These children are other proteins called RAB proteins. And she will bring them, say, to an enzyme, just like it's taking the child to school. And what that enzyme will do is it will change the protein. It will essentially put a little hat on its head. So if the protein goes to school, it'll graduate, it'll get the hat on its head. And then it will go out into the world, which is the cell still, and it will go there where it has to do something particular. And so all that Rep1 does, not all, it's very, very important, is to take these proteins around the cell once they've been modified so they can do something. They can do what they're supposed to do. So when we don't have CHM, we don't have Rep1. And when we don't have Rep1, we don't have these proteins where they're supposed to be doing what they're supposed to do. They're free in the cell and they're not really doing anything at all. They're unemployed. And this will cause trouble when there's enough of them. So, however, the cell can still function a little bit when Rep1 isn't there, because there is another graduate from the school, which is Rep2. And Rep2 is another nanny, rather, I should say. And Rep2 is still working in the cell, and it can still go around and take some of the other school children to where they're supposed to be. But even though it's throughout the body, it seems not to be able to do the same job in the eye. So this is why Rep1 is in charge of a very special group of children in the eye, in a group of these proteins, and Rep2 cannot teach them. So this is why we have the problem just in the eye in choroideremia. So one of the things we still don't know about choroideremia is, I've said to you that there are 3 tissues that degenerate. What we don't know is, does each one degenerate by itself, regardless of what the tissue next to it is doing? Or is there one tissue that degenerates first and causes the other tissues to degenerate? And this is perhaps a hypothesis we favor a little bit. It's that everything is happening in the RPE, possibly. And because it's in the middle, it's the sandwich filling. And if there's a problem with this filling, then it's impacting the tissues on either side, which is the photoreceptor and the choroid. And the RPE is a tissue that is very, very important in the eye because, as I said, its aim in life is to keep the photoreceptors healthy. And it will do this in a variety of ways, but every single key role of the RPE, that's its goal: keep the photoreceptors happy and keep the choroid also happy if it can. So that's what it's there for. So that brings me to the end of what is choroideremia. And just for the second part, I will talk about what have we actually done for this disease over the last few years. Not me, but in general, the scientific community. And so in order to be able to better understand the disease and also to test different therapies, the first step always is to try and do an animal model of a disease so we can really look and see what's going on. And the first animal model was created in the early 90s by Franz Kramer's group. And what they did was they basically just took the CHM gene out of a mouse because, like I've said to you, all the CHM mutations, what they do is they give no Rep1 protein. So if we take the gene out, then it's the same, there'll be no Rep1 protein. Only it was a little bit unexpected, but the mice actually died. And this was because we didn't know, but in the mouse, Rep1 had a very important role in the tissues surrounding the embryo. And so if Rep1 wasn't there, the embryos couldn't develop. So this mouse model was lethal. So then Miguel Seabra went on and he produced a very interesting mouse model, very complicated mouse model, where he inactivated Rep1 only in the retina, and not only just in the retina, but in the RPE and in the photoreceptors. So, and he could play with this to see whether it was the photoreceptors that were affected or whether it was the RPE that was affected. This mouse model was not lethal, and his studies went on to show that Both tissues seem to degenerate if there's a no rep 1, but it seems that the photoreceptors will degenerate much faster if the RPE is not in good shape. And then the last model that was identified for choroideremia was a zebrafish model. And this model is also interesting because it has a nonsense mutation in CHM. So then it's very interesting for that class of mutations that I spoke of with the stop sign. And unfortunately, this model is also lethal though, because fish, unlike us, do not have the rep2 protein. So when there's no rep1, there is no modification of these other proteins. So there's nobody around to do the job. So that's a little bit of a difficulty with it. But with zebrafish, it's a great model, is that you can actually use the larvae. And the fish actually survive for 5 days in the larval stage. So it's possible to look at the eye when they're in this stage. So they've been the animal models to date. But what's been, I think, very interesting model and rather a revolution throughout the last 10 years has been the possibility to generate human retinal models. And these retinal models are interesting because we can actually study the disease pathophysiology, we can test treatments really in the eye, in the cells of the patient that are truly affected, so really the patient's retinal cells. So how can we get these cells? Because we're not going to take them obviously from a patient. And the way to do it is via stem cells. And so in particular, I'm not talking about any stem cells. I'm talking about what's called induced pluripotent stem cells. So what does this mean? Basically, we can have a patient, say a CHM patient, we can take part of his skin and we can put these skin cells in culture and we can grow them. And then what we can do is we can treat them. And the factors we use to treat them with, we basically get the cell to forget that it's a skin cell, and we take it back to a stage when it was still really young and didn't know what it wanted to do in life. And that stage is called a stem cell. And so we take them from being skin, we take them backwards in time to being a stem cell, and this time we give them a new opportunity and say, what would you like to be now? And we would like you to be retinal cells. And then we can treat them and get them to become retinal cells. So we can do this as retinal pigment epithelium. And even more recently, and this is probably the last 5 years where this technology has been really advanced, we can make what's called organoids. And organoids are little 3D structures that can mimic the retina, and they have all the photoreceptors lined up on the outside. So today, we can use RPE and we can use photoreceptors of retinal patients. And this is what we use in our lab. This is basically now we hardly use any mouse models and we only use human retinal models. So as these models contain the gene defect from the patient, from his skin cells, we basically have reproduced the retina of the patient. And therefore, I can hear myself. Can everyone? I mean, I can hear an echo of myself. That doesn't matter. And so now these become a very powerful model to either test genetic mutations, to try and understand pathophysiology, but also to test the efficiency of therapeutics. So what is the therapy that we've advanced with the most in CHM? And that would be what is called gene therapy, but what is more precisely called perhaps gene supplementation or gene replacement. And the concept of gene therapy is to put a gene back into the cell, a healthy copy of a gene back into the cell. So the CHM gene, it's mutated. We bring in a healthy copy of this gene. However, to do this, we can't just put DNA in any cell. We need to put it inside a vehicle. And the vehicle that we use mostly in gene therapy are viruses. But what we first do is we take everything out of a virus that can make us sick. And all we put in is the gene that we want to put in. And then these viruses, they still keep their ability to enter cells. That's what viruses know how to do. They enter cells and they make us sick. And so in this case, the virus can still enter the cells, but this time all it's going to put in there is the gene that was missing, the CHM gene. And in this way, we really treat the cause of disease. So gene replacement or gene supplementation is very interesting because it can treat any, regardless of the mutation. And so for CHM, there have been a few gene therapy vectors, so that's what we call once we change the virus, that have been tested in patients. And before we can test them in patients, we need to test them in animal models. And so the mouse model was used to test two different, glasses time again, two different vectors. So the first was tested in 2013. It was by Miguel Seabra and Rob McLaren. And it was, what I should say is, first, I'll go back a step. I said we use viruses. The viruses we use the most are called AAVs. And we use these because they're small, so they can get into cells even easier. And they don't make us sick, usually. So it's going from bad to worse, isn't it? Okay. Oh, that's better. So these AAVs, there are lots of types of AAVs, and the very first AAV to be tested was called an AAV2. It doesn't really matter, it was just the type. And also, quite early on, Jean Bennett tested another AAV called an AAV8. Then we went on, and Jean Bennett also tested another AAV2 vector, very similar to what Rob McLaren had used, just a little bit different in what she put in it. And she did this in patient stem cells. And then using patient iPSC-derived RPE, we had actually tested an AAV5 vector for CHM. And more recently, in 2018, Jason Mills, I think working with Jean Bennett still, tested a new type of AAV, an AAV that was made more so. It was identified, it was very different in its coat than the other ones. And this was also tested. And essentially, all these vectors showed that they could reintroduce CHM gene in cells. But only two of them went to the clinic, and they were the AAV2 vectors. And the first was the one of Rob McLaren, and that actually went as far. It was a phase 1/2 trial by Nightstar, and then a phase 3 trial by Biogen. So it was tested in even more patients, and that's the trial I think almost everybody knows about today here. And there was another trial, which was the one by Spark Therapeutics, which began, I think, in 2015. And for now, as far as I know, at least the results haven't come out from that trial. So of all these, only AAV2 was tested in patients. So this is really where we are today. And so where are we going to go tomorrow? And it's really not finished. We always feel that once there's a clinical trial that it's all done with, and it's really not. I think research never ends. That's what's hard for us researchers, in fact. It's just constant questions coming up, constant ideas, constant improvements. And so I think a lot of the questions we're asking ourselves now with gene supplementation is, are we doing this the best way? So with gene therapy vectors, with the AAV vectors, if we want them to reach the photoreceptors and the retinal pigment epithelium, we want to put them in what's called the subretinal space. And this space is situated between the photoreceptors and between the RPE. We inject in this space, we separate them temporarily, we put the fluid containing the vector in there, and it is absorbed by the photoreceptors and the RPE. And then the space closes, usually without any problems. But you can imagine that if we have a retina that is already relatively fragile, it's always a risk when we have to separate it, and we don't always like doing this. So these surgical complications are pushing us to think maybe there's a better way. And there is another way, which is called an intravitreal injection. Now, I told you we had the retina at the back of the eye, but inside the eye, then there's a big cavity, if you like, and this is the vitreous. So you can see that the retina, the front side of the retina, is also in touch with this cavity. So one other possibility is to put gene therapy products here in this space. And what we then would hope is that it can go through those neuronal layers to get back to the photoreceptors. Essentially, it'll go the same direction that light goes. So there are some efforts now to try and develop vectors that can go in this way, through this space. And this is the work, or this is the clinical trial that 4D Molecular Therapeutics is doing. What they basically have done is they've identified a new AAV that is capable of crossing all these barriers, because generally, really, our bodies are always very well done and they're there to guard against invaders. And so all these retinal barriers are put in place so nothing can get into our eye. And what we're essentially trying to do now is go through them. So it's very difficult. And so 4D Molecular Therapeutics have identified an AAV that has the capacity to do this, to go through these retinal layers and reach the photoreceptors. So this is a clinical trial that is ongoing, and I don't believe we have the results yet, but it is in progress. However, as I said, the eye has put these barriers in place for a reason. And if we put a foreign substance in there that goes through them, um, we can imagine that perhaps it also could bring up safety problems. So there's also a lot of work going on to identify maybe other vehicles, not necessarily coming from viruses, but different ones, such as lipid vehicles, that can bring DNA into the eye as well. And there's a lot of work being done with things called nanoparticles, and they can be administered from the subretinal space or the vitreous, and they need to be optimized. And I think you'll be hearing a lot more about this tomorrow from Richard Hardbottle in his talk. So what else is possible? And as I said to you before, there is this big class of molecules, big class of mutations that are nonsense mutations, with the ones that bring the stop sign. So there are actually drugs that we can administer and that will sort of hide the stop sign from the cell. Essentially, they'll take it out. The only thing they can't always do is put the right thing back in. So generally, if they can hide the stop sign, the cell can make a protein. Most of the time it might be fine, and sometimes maybe it'll be a little bit different. But the idea is just to be able to get some more Rep1 in there that's working. So this is a possibility on these with these molecules. And what's good is there's the zebrafish molecule that has this nonsense mutation, so it's good for screening such molecules, and also the patient RPE that is generated from patients with nonsense mutations. And Maria Masagi did a study a few years ago, and we collaborated with her, where we tested a couple of molecules in the zebrafish and in patient RPE. And what it seems that these molecules maybe work, maybe depends the type, but their efficiency also might depend on the type of nonsense mutation. So maybe we can't say we can treat all of these nonsense mutations, but there are maybe some that will be in a genetic context that could be efficient. And so Maria will probably, I think, talk more about this tomorrow. And just one other thing I wanted to say about this was that, again, these mutations put in a stop sign. When the cell reads the DNA, gets to the stop sign, it'll produce the RNA, but the RNA will also be shorter. And so what Maria's seen recently is that there are ways to maybe increase this, because when the RNA is shorter, the cell wants to get rid of it. The cell is very fussy. She wants things perfect. In quality, she has high expectations. If something is shorter, she gets rid of it. So what Maria's been able to show is that there are some molecules we can treat cells, and we can increase this RNA, even if it's shorter, and then we can increase the efficiency of these drugs to give enough Rep1 protein. So there's still work to be done with these molecules. So there was the gene replacement. There's maybe pharmaceutical or drugs that could work. There is also perhaps another approach, which is called gene independent. And this is an approach that wouldn't be just for CHM, but it could be maybe for all retinal diseases. And it's a group in Paris that had identified this factor, and it's called rod-derived cone viability factor. And it's actually a factor they've identified that is produced by rods, but to protect cones. So the rods actually produce this factor, and it's important for cone survival. So this may be a reason why when we have rod cone diseases, the rods die, but then the cones die, and maybe because the rods are leaving and they're taking with them some things that the cones need. So what Sparring Vision is trying to develop is a way to introduce this factor into the retina, probably with gene therapy, putting it into other cells in the retina that can be a reservoir to produce it. And then it can be used to preserve, for example, the cones that are remaining. So it would be a way to just preserve the central vision for as long as possible. And then another revolution that's happened, I said the first revolution was the stem cell derived models, and the second revolution of the last 10 years has been what's called genome engineering or gene editing. And this is something that instead of this time trying to put a gene or DNA into a patient's cell, what we try to do with gene editing is go to the patient's DNA and correct it. So, what happens is it's a system that will cut DNA a little bit near the mutation, just a little bit. But again, the cell doesn't like cuts in her DNA. So, she will come and she will fix it. And so, in a way, we will force the cell, if we can, to fix it to read the right sequence. So it's a way of erasing a spelling mistake and rewriting everything correctly. And so this has a lot of potential for genetic diseases, not only for repairing mutations, but also even just for disease modeling, because you can imagine if we can fix a mutation, we can also introduce one. So it's a way to generate animal models much easier. And I think David Gamm may talk more about this tomorrow. And then lastly, so these these um, these approaches that I've mentioned, they can help a retina that is not functional. But as disease evolves, if the cells will start to die, and whatever we do, if the cells are dying, then we can't put in a gene and help them recover. And the thing to do then is to try and replace the cells. And that's another reason why the human stem cell models are so important, because they are a source of retinal cells for transplanting patients eventually. And for example, human RPE transplantation is actually being done in the clinic. So we know for now what we can do, what we can't do, what we need to address, what we need to improve. But it's actually come a fair way. And photoreceptor transplantation is not yet there, but there are a lot of studies being done in mice to try to understand what stage we need to take these baby photoreceptors and put them in the eye for them to develop well. So this is still in research stage. But the creme de la creme really would be if we can combine genome editing with stem cell transplantation, because then what we can imagine is we will take a patient's cells, we will make them stem cells, we will correct the mutation, we will turn them into retinal cells, and then maybe what you can do what is called an autologous transplantation. So we put the retinal cells back in the patient's own eye. And this is really interesting because it would there wouldn't be risk of rejection with immunological responses. And I believe that Brian Balios will talk about this tomorrow. No, Saturday. And so I'll just get to the end, which is this is what we know, this is what we've done, this is what we're trying to do. But with all these therapies, I think one thing we mustn't forget is that we don't know everything that's going on in CHM. And we know very well the gene, we know very well the protein, thanks to Miguel. We know very well what the protein does. What we're still missing is how do we get to the phenotype? Why is it that the cells are dying really? And that's what we're not sure about yet. And so what we're doing is now trying to understand the pathophysiology of the disease with the iPSC derived models like the RPE. And I've said it before, but I'm such a fan of this tissue, I'll probably say it again, is this tissue looks exactly like the tissue in the eye. Exactly the same. It's the cells are standing up like they should. The cells express different proteins where they should. And above all, this tissue, it reproduces every single function that the RPE does in the eye. And we've set up a whole panel over the years of tests to analyze each of these functions, and it does them all. And so we can imagine that then we can see what's going on. And I just want to finish with this one small example that to give you an idea where our research is going, is one of the functions of the RPE is to secrete growth factors, and it will secrete a certain quantity of growth factors towards the photoreceptors and a certain quantity towards the choroid. And this is a very fine balance so that everyone's happy and healthy. And what we've seen in the patient RPE is that there is a reduced secretion in both directions of both towards the photoreceptors and towards the choroid. And so it's beginning to give us a clue of what could be maybe not working correctly. And this could explain also why both tissues are then degenerating each side of the RPE. And this is probably the most complicated I'm going to get, but um, underneath all these roles of the RPE, it's all finely regulated in the cell by different things, and it's in particularly finely regulated by ion flow in and out of the cell. So things coming in, things coming out. It's very important for the cell to work correctly. And what we've seen recently is that perhaps in CHM patients, things aren't working well in terms of calcium channels. So these are channels that bring calcium into the cell. And what we find is that the cells bring it in, and in healthy cells, we can add a inhibitor and say that's enough, don't bring any more, we've got enough. But with the CHM cells, we can't tell him to stop. They don't want to stop. They keep bringing it in. And so we're trying to explore this process at the moment to see what is going on, why the calcium channels are dysregulated. And it's important because then it can maybe give us clues to other therapies. So I thought I'd finish, but I hadn't. One last thing is that I mentioned to you these organoids, and these organoids are really beautiful. And for those who have access to it on the slides, the green cells around this round structure are photoreceptors. So again, in the organoids, the photoreceptors are exactly like they are in the eye. And they're standing up like they should, they're organized like they should, and there are both cones and there are both rods, and the cones are less abundant than the rods. So what we're trying to do also is with the photoreceptors of the patient, also see if we have the same defects that we have in the RPE and in the photoreceptors. And maybe in this way we can better also work out which tissue is actually playing up. And I think the future really is these stem cell models, always in combination with other models, because it doesn't reproduce a whole body, but there's a lot we can do, we're realizing now. And I think a lot of work's going into now we've got photoreceptors one side, we have RP the other, is how to grow them together and how to make them grow together and then perhaps respond to light together. And that way this will bring an extra dimension of working out how a tissue is functional and seeing if drugs can make it functional. So to conclude, my first CRF conference was in 2008, and I think when I look back over the last 14 years, it feels like everything's very slow, but there has been so much tremendous stuff that has been done. And so, for example, there have been 3 products in clinical trials for CHM, 3 gene supplementation products, and this is pretty much unheard. I can't think of another disease where this has happened. It's usually we go with one and that's it. But there's been 3, and although we're at the moment a little bit disappointed, I think we should really keep that in mind. It's quite a feat. And I think these, again, the revolution of the stem cell models is bringing up, also is opening all sorts of perspectives for the future, as are all the new therapies like the genome editing, as well as all the other therapies that are now emerging that we're realizing maybe we can do this. So there's still a lot of hope for the future. And also that all the work now going into better understanding CHM perhaps can also bring up things that we haven't thought of before. And so to finish, I just wanted to also say that for us researchers to work with patient associations it's an indispensable partnership. And we really, I'm convinced, none of us would have gotten so far for CHM as we had today if it hadn't been for all your support. So thank you, and thank you for listening to me.
This on? Oh, there we go. So first and foremost, thank you very much, Dr. Kalatzis. That was outstanding, number one, and you did just a great job working with our with making this a challenge for you, and we're very sorry about that. We have a few minutes left, and we have a couple of gentlemen who have microphones. If someone has a question or two for Dr. Kalatzis, if are you willing to answer a question or two?
Go ahead first, see if the mic works.
It works. Maybe a simple question, but in terms of gene therapy and the different mutations, if gene therapy works, it's in phase 3 and it's approved, is it applicable for all mutations, no matter which variant?
In CHM, yes. Alors, if we talk more generally outside of CHM, no. Where there's limitations for gene therapy is with dominant disorders, because as I said, there's this toxic protein that even if you bring more healthy protein, it'll be a problem. For CHM, there aren't very many what's called missense mutations, which are mutations where the protein is still there, but a little bit altered. Because if that protein was there, maybe we can imagine that if we bring another one in, it's going to interfere with it, compete with it in some ways. But we did a study a few years back and where we showed of a missense patient that gene therapy in the case of that patient as well was fine. So no, in theory, all the loss of function mutations should be treatable by gene therapy.
Do we know from the clinical trials if there was a difference between different mutations from the patients that got treated?
Yeah, I don't think so. The trials, they never looked, I don't think, at each particular, correct me if I'm wrong, but from what I've read, they never didn't do that correlation. I think to do a correlation like that, you'd need a lot of patients. So I think it's more patient versus control than with the number of patients that are treated. Can we divide them up into categories and look what's better and what's not better? So,
Thank you so much.
okay.
Do we have another question?
Is it on? Okay.
Mm.
Dr. Kalatzis, very nice to see you again. I feel like I should know this, but in the gene editing, if that does correct it, does that pass on to your offsprings if the gene's
No.
corrected using that process?
Yeah, no, it's not the germ line that we're working with. It's just somatic cells that we're correcting. So no, there's no transmission.
Okay, thank you.
You're welcome.
The other guy, Neil, is my daughter, Callie. She gave me a look when you said it was a guy. She was very insulted. Any other questions?
Sorry about that. Do we have another question?
Thank you. How long? My grandson's 6 years old. Do we have some idea when we'll have an answer?
In terms of therapy?
Yes.
Yeah. I think right now the big message is we need to be patient and we need to really see what's going on with the Biogen trial and the Biogen results. I feel that Biogen set their criteria for saying therapy has worked very high, too high, and I think we've really got to take the time and they have to go through the data and they really have to be because what's really it's great for patients but it's difficult for a trial is that the disease progresses slowly, so they really need to give more time to look at the results. So I can't give you a time, but what I would like to say is just for now, for 1 or 2 years, we just have to be patient for these results to come out and then see where we're going and where we're heading and what's possible and what's not. Because if we speak to patients, they're very positive, or the patients we've spoken to anyway.
Thank you for taking that question, Dr. Kalatzis. I I guess we had a pool to find out when that question would first be asked, and it probably
There's
will
the
be
winner.
asked again. Just so when are you going to fix everything? You know, no big. We can take one more question, and just before that, I'd just like to say again, thank you very much to Dr. Kalatzis. The CRF has had a relationship working with Dr. Kalatzis since 2008. She first attended one of our conferences in 2008, and we have been funding various research projects in her lab with her work over the years. She's currently one of our active projects, and we of course appreciate everything and anything that you do for us and for other diseases also. We feel that possibly, you know, breakthroughs in other diseases could lead to helping with choroideremia. So if we can do one more question, and then we're going to move on to a recognition of Dr. McDonald.
Again, thank you so much for all the information. This is it's amazing, and I was going to ask the same question the gentleman back here asked. I have a 5-year-old grandson who's affected, but and this may be super still stupid, but I'm assuming that funding is what really the more funding you have, the faster things could happen. And I understand some of it you just need to wait and be, but I guess the bottom line is the bottom line. You know, if you had more ability, then could things move quicker?
I, yes, of course. Funding always helps things move faster. Always. But there's always also, like, for example, the work we do, the models I didn't say, but like the photoreceptors to develop, they're 9 months. I mean, 9 months for a photoreceptor that looks like what we have in the adult eye. So there are some things that are not compressible really, regardless, and also, yeah, it's also training. We also, at least in France, we're not allowed to keep people very long term. So we then have to keep training people. So it's always things like this too that slow us down a bit and we can't do much about. But yes, unfortunately, money makes the world go round, doesn't it? So yeah, thank you.
All right. Thank you very much, Dr. Kalatzis. That was just a stellar moving around by Dr. Kalatzis. As you all can tell after that presentation, we were unable to bring Dr. McDonald or Dr. Ciabra into the presentation, and initially Dr. Ian McDonald had intended to be here, and he's very disappointed that he couldn't make the conference. We want to take just a couple of minutes before lunch in recognizing Dr. McDonald. He's been involved with the foundation for many years. He's been on our science advisory board for many years. He's been the chair. He's a virtual institution. I have not known him all that long, quite honestly. Dr. McDonald, in his spare time, is an artist, and we thought it would be useful if we were to honor him while we're here and present him with a gift, a small appreciation of our thanks to him. And we came up with this artist box. It's a lightweight travel box, and on it there's a little plaque saying Dr. McDonald, Choroideremia Research Foundation, Science Advisory Board Emeritus, and the CRF Community Thanks You 2022. So next to me is John Oster. He has known Dr. McDonald for many, many years, and I asked him if he would just say a few things about Dr. McDonald in appreciation of what he's done for the CRF.
Thank you, Neil, and I'm happy to speak about Dr. McDonald. You know, my brother Matt and I started attending conferences of Foundation Fighting Blindness in the mid-90s. And I honestly can't remember if it was Chicago or Los Angeles where we met Dr. McDonald. It was one or the other. And by then he'd already been working on choroideremia for probably 15 years. A wonderful guy. He had lunch with us, which he certainly didn't have to do. We had a great chat. We talked about lots and lots of things. I think it's fair to say that his commitment and compassion and persistence is what gave the rest of us the inspiration to start the foundation and keep it going all these years, and look for great things in the future. So thank you, Dr. McDonald. I think we all hope to see you real soon, and everybody in this room owes him a great debt. So thanks.
From me to all of you, thank you very much for the honor of receiving this gift virtually, and I'll put it to good use. and we'll see what I come up with.